Hello, and welcome to the Field Guides. I'm Steve, and I'm here with Bill and Alex.
Hello, Steve.
Good afternoon, guys.
Good afternoon.
Hello.
What we're going to do today, and over the course of many future episodes, is give you the experience of what it's like to be out in the field, in the woods, and on the trail. For every episode, we pick a natural history topic, research the science on that topic, head out to a natural area, and share with you everything that we learned. And today, as you may have noticed, we're here with Dr. Alix Matthews. And she's a postdoctoral research associate in the Department of Biological Sciences at the University of Buffalo in Buffalo, New York, and holds a research associate position at the Buffalo Museum of Science, which I did not know that.
Wow.
Very cool. So Alix is an expert on the ecology and evolution of host-symbiont interactions. And speaking of that, well, Bill, do you want to say where we are first?
Yes.
Yeah.
All right, so we are in for the first time on Mike, Walton Woods. And this is a 50-acre park. It's in Amherst, just outside of Buffalo, not far from UB. And it's managed by the town of Amherst, so this is a town park. And there's woodlands here, there's wetlands, there's open water. Most of the trails are paved, so folks, we're not going to have a lot of foot noise.
We tried our best in the beginning.
Yeah, that's right. But the trails circle both Walton Pond and Lake Audubon. And all the habitats here, we're going to find lots of deer, as well as...
I was seeing squirrels, rabbits, everything.
Yeah.
The crows are going wild, a million robins for some reason.
some blue jays in the
Yeah.
background.
Yeah.
So there's going to...
Osprey.
Yep.
Oh yeah, the osprey when we came in.
Yep. So there's waterfowl, songbirds, and this is an active park. So just like in many of our episodes, when we're in a town park, a little bit more suburban area, we're going to probably be running into other people. So folks, you may hear people walking by, joggers,
Might hear us
loud,
trespass onto people's backyards by
possibly,
mistake.
possibly, but
It's a nice area though, and even though this is much closer to roads than normal, I actually think it's better than some other locations we've been to in terms
I
of
agree.
the road noise.
at least right now it's relatively
Yeah.
quiet.
Yeah.
And we are recording right now. This is what, September 4th?
4th.
5th? September 4th. So this is early September here in Western New York. Hopefully we might hear some migrants moving through, but
Oh yeah.
since the breeding season is over, it is pretty quiet right now. We're not hearing tons of bird sounds.
I just heard like 400 million birds were like starting to migrate, or it was estimated like 400 million on the radar just like yesterday or 2 days ago or something.
Yes.
It's a big
Yeah.
push right now.
Yeah.
So any listeners that don't know about it, there's in recent years, there's lots of people that are posting on social media about how weather radar is being used to track bird migration in the fall and the spring. So it's a whole rabbit hole you can go down,
Yeah.
and it gets kind of addicting to see how big is the cloud of birds rising up each evening, right? All right, but we're not here to talk about birds. At least,
well. Well.
but we are actually.
Sure.
All right, so Steve, the last thing that you had said.
I was about to say
Yes.
that, Alex, maybe do you want to give us a little intro onto what the topic's going to be today?
Yeah, so I am a feather mite biologist and an ornithologist. So I study pretty much everything that you can think of about mites that live on the feathers of birds. But I also study avian ecology and a little bit of avian evolution as well. So that is what our topic will be today.
And when you were growing up, did you want to be a feather mite biologist?
I had no idea I could.
This is great, though, because I don't know if you know, Steve and I, we bird band. And we often
Well,
will
one
find...
of us bird bands more than the other one. So, meaning that I haven't been there in a few years. So.
Yes,
Yeah.
but you have two kids. So.
Yeah.
But we often find feather mites
Yeah.
on our birds that we catch. I know nothing about feather mites, but I will say that one of our volunteers is the vet from the Buffalo Zoo, and he is often very interested when we find some feather mites, and he wants to take pictures and figure out what they are and what they're up to. So
Wow.
Absolutely.
yeah. So can you tell us, like, how did you come to study something that I'm betting a lot of our listeners have never heard of?
Yeah. That's kind of how it started as well. My master's advisor, he and I were working on trying to get a project together for my master's thesis, and we had one idea in mind that kind of all fell through. But then, so we just kind of had to come up with a new idea. And he said, I have some feathers in my office that have these little bugs or something on them, and I don't know what they are. Do you want to try to figure out what it is? And I was like, sure, that sounds great. I had done some undergrad work on avian malaria, so I had my toes dipped into sort of the parasite, symbiont, pathogens world, and I was like, I could probably ask similar questions that I have been studying in avian malaria with feather mites. Well, at the time I didn't know what it was, just these bugs on the feathers. And turns out we just kind of built the project from the ground up, and that's how it got really started. Is just something that you know, as biologists we like to collect things and keep things in our offices for really unknown purposes, maybe for future projects.
You never know.
Right.
And that's exactly what happened. He just had all these feathers in his office and was like, let's try to figure out what's going on with these bugs that are on them. They're not really bugs, but that's.
So before studying feather mites, you were studying birds.
I was studying birds,
Okay.
yeah.
And is that something you grew up like with a passion for, that you wanted to study?
I liked birds. I was always fascinated by them. I was, enjoyed watching them. My grandparents had a lake house, and so we would go to the lake, and it would be really fun to watch the waterfowl. There were like really cool nightjars there, so it was just like a neat. I liked birds, but I didn't know I could study them until I got to college.
For the listeners.
Yeah.
Let me interrupt you for a sec.
Yeah.
What are nightjars?
They're a group of birds that are more active at night, typically. Yeah, they're a family of birds.
It's a misnomer because you would think they're jars.
Yeah, they're not jars.
But check them out, folks. Very, very interesting birds.
Yeah. Whippoorwills too. They would call all the time. So those were the birds I became most interested in. And woodpeckers too.
So,
but I don't study either of those now.
Oh, I was going to ask.
You've moved on.
Yeah.
Okay.
Cool. So let's start with the obvious question. Are birds real, or did the U.S. government exterminate all birds between 1959 and 1971, and then replace them all with surveillance drone replicas, and then they sit on power lines to recharge, and then they poop on people's cars to track civilians?
I can't talk about that.
Okay, I knew it.
Is that really part of the conspiracy, that the U.S. government killed all the birds?
Yeah, but I think it's like a fake conspiracy. I think it's like a satire conspiracy.
Yeah.
Okay, all right.
Yeah.
So hopefully people aren't actually thinking that. I think I did know that.
Yeah.
All right.
Sorry, I felt like I had to ask that.
That's a good. I think the person who started that is from Memphis, where I used to live. So, yeah, there was big billboards all over town that says birds aren't real.
Yeah,
right.
Kind of blew up.
It's whoever it is, they make a lot of money now from merchandise, selling merchandise.
From Birds Aren't Real?
Mm-hmm. Yeah, there's a whole website.
I kind of want to support it.
I know, right?
Yeah.
We missed the boat there. All right, Steve, any real questions?
Okay, so the first thing that I started to think about, first of all, I want to say the reason that I invited Alix on the podcast is because I think it was it last semester or the semester before that you gave that talk to the department?
Maybe it was, I don't remember now.
Yeah,
I think it was the fall semester.
Okay, last
year.
fall, I just, I work as a postdoc. I don't really go to a lot of talks. Maybe I should go to more than I do. But we had someone in our lab giving a talk on the same day, and Alix was also giving a presentation. And her presentation was all about feather mites and some of the research that she was doing. And to be totally honest, I had never thought about feather mites before.
Likewise.
And as like a fellow bird person, I was like, this is, it was pretty exciting to me. And it was just a whole interaction that I'd never thought of before. And then it took another almost a year to ask you to come on. So...
But I will vouch for Steve. After he saw that presentation, he did say, hey, I was really impressed by this presentation. We need to have her on the podcast.
And here we are.
And then I finally worked up the courage to say something. All right, so...
And Steve does not hand out compliments very easy. So.
No, I'm usually like, this person sucked, they suck. All right, so when I think of mites... when I think of our tick episode, because ticks and mites are part of a huge group, really diverse.
Yeah.
I think there's like tens of thousands of species of mites. They're huge. But I remember the one little biology factoid I knew growing up was that humans have mites on their face.
Yes.
And I'd happen to know the name for it because maybe it's got a ring to it or something, but it's Demodex follicularum.
Yep.
Right? Mm-hmm. Now, there's another one, Demodex brevis, I guess, that's also common, but I don't really know much about it.
Yeah, I can't remember what that one...
Whoa. I think a tree might have just come
Oh,
down.
there's a deer right there, too.
Oh, yeah.
You're looking for a deer.
Oh my gosh.
Yeah, we got a deer right there.
Folks, there's a deer about, what did you say, 50 feet off the trail?
Yeah.
Just watching us.
So, okay, so the Demodex follicularum,
Those are
they're-
basically follicle mites.
yeah, they're just living on your face. In fact,
Yeah.
it's not just fine that you have them, it can be beneficial that you have them
Yeah.
as well. Now, something like scabies mites, that's a problem. That's like, that'll give you rashes, dermatitis, things like that. Now, when we think of feather mites, should we be thinking about them as Demodex follicularum, or should we be thinking about them as scabies mites, or maybe a little bit of both, or maybe something that I haven't even considered yet?
That's a great question, and one that feather mite people are arguing about and trying to come up with solutions and answers to. We don't have a super good understanding of whether they are parasitic, so causing harm to the bird. Probably not. A lot. Most evidence is not super strong towards parasitism, but whether they're mutualists, actually helping the birds, there's a little bit more evidence potentially suggesting that they could be helping the birds. But I'd say the majority of research suggests that they're commensals, so they're like just living on the bird feathers and not really doing a whole lot. But
So they're benefiting, and the bird isn't necessarily suffering
harmed or helped,
for anything.
Yeah.
Can you give the audience just an idea? So feather mites, when they're on the bird, like where are they living? What exactly are they doing? Why are they there?
Yeah. So the ones that I study live on the flight feathers, so on the veins of flight feathers on the wings and on the tail. And they can live on either the ventral side and kind of tucked in between the barbs that are holding the feather structure together and keeping that feather nice and strong so the bird can fly.
What side's the ventral side?
The front side.
Okay.
Like your chest.
Yeah,
Vent.
Yeah.
the underside.
Dorsal's like a dorsal fin, right?
Yeah.
Yes, exactly. And there are feather mites that live on the dorsal side of feathers as well, but their body structure is completely different than the ones that live on the ventral side, just because of the aerodynamics and the
exposure.
The exposure, yeah, exactly.
Oh, that's so cool.
Yeah, and there are other types of feather mites that are living on closer to the skin, closer to the quill. There's also quill mites that are sometimes grouped into feather mites as well because they're associated with the feather. Just depends on like how specific you're being about the taxonomy. But so there's mites that live inside of the quill, mites that live on top of the feather, like the ones that I study. There's ones that live kind of in downy feathers, so the insulating feathers closer to birds' bodies that aren't as
long and
not as
structured. Yeah.
And then there are nasal mites that live inside the nares of birds. There are lung mites.
Whoa.
There's mites everywhere on a bird.
So
I want to jump in though. So this is going back to something that Bill said now. You said that when you're banding, people are excited, or at least one person is excited about the mites.
At least one person.
Yeah, at least one person's excited about the mites. Does that mean that they're visible? Well, it also goes back to what you said. Is how did your PI know that they were on the feather? Are they so big that you can just see them with the naked eye very easily?
Yes.
And is that also what you're seeing at bird banding?
Typically, that's probably what you're seeing when you're bird banding. Probably the same ones that I study because they're the most obvious. When you, if you're measuring a wing, you can. They're microscopic, but they are visible to the naked eye. They look like, flecks of dust.
They're like a fraction of a millimeter, right?
Yes. Yeah. They're very small, but they're still visible. They just look like little specks of dust.
Yeah.
I'm curious what the variation is between like the biggest and the smallest that actually like live on birds. You know, like anything from something we just couldn't see with the naked eye to something that
that just covers the entire bird.
It's just riding it, like
Yeah, there's some genera that are much smaller than others, but those are still like visible. You can see them, but you might be confused on like, is that a speck of dust or is it an organism?
Because I'm not very observant, and when I probably saw feather mites many times, not realizing they were feather mites until Erin Carnatz, our bander, who's our master bander, she has a permit. She's the one who pointed out, oh, those are feather mites.
Yep.
I'm like, oh, those things are alive. Okay.
Yeah. That's the same thing with my PI that saw them, we had no idea we didn't know if they were lice or if they were ticks or what, who knows, no idea what they were.
So I did a tiny bit of reading before we got here today, and am I correct? And there's thousands of species, right?
Yes.
So I imagine there's a lot of variability in terms of size and where they're living and just what they're doing on the bird.
Yeah, absolutely.
Okay.
There's approximately 2,500 species of feather mite, the ones that live on the veins of the feathers, and then I think that's about estimated to be about less than 20% of estimated biodiversity, which is not very much.
Do you know, is there a similar amount of species that live on people?
I don't know.
Okay.
I don't know.
We'll have to put that in the episode notes.
It's always about people with you. Like, who cares about people?
Very
We're talking
anthropocentric.
about birds right now. Although I did hear that people, when I was looking at what mites that can affect people negatively, like obviously the scabies one came up, but then I also heard something like bird mites can get onto people.
Is
that a worry in your lab at all? Or?
No. The ones that I study are not. They're so specialized to feathers that if they do, I've gotten them on my hands and stuff before, but nothing happens and they just kind of flake off. But chicken mites, I can't remember the species name, but those ones, they are. Those ones can
I have chickens.
No.
So I should check them when I get home.
Those are the ones that can get on humans and cause irritation.
Oh, okay.
I sort of felt that about you, that if someone I knew was going to have mites,
kind of gross.
The chicken guy.
It's not deadly or anything, is it?
No.
Okay.
It's very itchy.
Okay. All right.
Deadly.
How'd he die? Chicken mites.
Chicken mites. I got him.
So with this wide range of species, am I correct? When we ask a question like, are they beneficial? Are they harmful? I got to imagine that, it depends on what species you're talking about, right? Yeah. So it's hard to make generalized statements about 2,500 different species.
It is, and I think that's probably why we haven't found haven't found a really strong pattern either way with beneficial versus parasitic, or maybe they actually are just commensal and kind of not really doing anything, but maybe because there's so much variation that it's all just kind of evened out by studying so many different species. I think that could have something to do with it.
So what are they looking for? When they're on the bird, or they're living in the feather and the lung or the nares, what are they looking for?
They're looking for space and food, and I mean that would probably be about all that I could say.
Are they eating the feathers themselves, or are they eating what's on the feathers?
They are eating what's on the feathers.
Okay.
Yeah, so they eat bacteria and fungi and plant matter and diatoms and oil, potentially oil that birds are taking from their preen gland and spreading onto their feathers. So there's been some microscopic images of big globs of oil inside the bodies of mites that are mounted on slides.
That's
and
cool.
so we know that they're eating it, but we don't know how necessarily how often they're eating it, or if they're eating the stuff that's inside of those oily globs.
So is it random or non-random, or because of something else they're going after?
Yeah.
So there's a lot we don't know.
There's a lot we don't know, which is
Job
the beautiful
security.
thing about getting to study these really interesting creatures that we don't know a lot about. It's like anything you have a question about, it's probably not been asked before. So,
Well, so one thing that I did very little research on this because I knew we were going to have an expert today. But I read about the same thing, that there was 2,000 or more species and about 33 families. But then when I think about the biodiversity of birds, there's a lot more birds. So there's I think there's a known 1,000 bird species, and that's in 250 families. And not that the number means anything. Maybe we're just splitting them up more, arbitrarily. But there's a lot more birds than there are these mites.
Right.
Is this something that can we expect? With people, we just assume all people have mites. Do we also assume all birds have mites? Is this just a normal thing? Is this what it means to be a bird, is to have mites? And
no, that's a good question, and that's the 2,000 to 2,500 species is probably an underestimate because we haven't sampled every species of bird that's out there. for every possible species of mite, and each different, there's, like I said, there's ventral mites, dorsal mites, mites in different like areas of the bird body. So it just depends what type of sampling you're doing as well.
Okay. Well, so there's all this diversity.
Yeah.
Would you, maybe instead of asking a general question, could we hone in on something that you've experienced? Like, let's say on a specimen that you're looking at, what's the diversity like on a single bird that you've studied?
Yeah, that's a cool question. So for the general group of feather mites, the feather vein dwelling feather mites, on a single feather, I found 4 different genera at once, which was really bizarre. That usually doesn't happen. It's usually 1 to 2.
It's a very cosmopolitan bird. It gets around.
Yeah. Which was really, that was really neat to see that. It was like, oh wow, there's so much happening on. I think of like the bird as an ecosystem in and of itself, like an individual bird, but thinking about, okay, this one feather on this one bird is harboring all this diversity. How is that even possible? So, that's...
Now, when you talk about the 4 different genera, just for this example, and it's okay if you don't know it, I wouldn't expect you to know all these things. Like, how distantly related are they? Are they like their populations diverged tens or hundreds of millions of years ago? You know what I mean? Like, I don't know
about the timing for sure, but the species that I found, 2 were in the same family. And then everything. So there were 3 different families represented on this one single feather.
That's got to be relatively
They're pretty
ancient timing.
Pretty distant.
You would think.
Yeah, pretty distantly related. That's relative. But.
Because I typically work within angiosperms. So with a lot of the stuff that I work on, it's like most of the time you're only going back like 120 million years. You know, that's when that was like the ancestor of all, at least the core eudicots. And then now with fish, it's going back a little bit further, like 350 million years, for example. But with mites, I have no frame of reference of what timing I should be thinking about.
Well, for feather mites, they, there's no. Not that I can remember that there's any fossil, like they don't fossilize, but
it'd be a tough find.
Tough to find.
They fossilize really well. No one's looking for them. So.
But their like rapid diversification on feathers happened approximately the same time that feathers evolved, which was about 150 million years ago.
Okay.
With, they kind of like set it to Archaeopteryx, more or less. That's kind of like the
Are there scale mites?
Like on reptiles and stuff? Yeah, there are mites that live on reptiles. I don't know if they are scale.
Because feathers are specialized scales, right?
Yeah.
So it makes sense. Yeah. So I think I need to stop us all for a sec, because I think one important question that many people listening right now are probably wondering is, what does a feather mite look like?
Oh, do we want to walk and talk about this?
Yeah, let's do it. Yeah.
So they are specks of dust. That's to the naked eye, they look like specks of dust. They have 8 legs, so they're arachnids. So they are related to spiders. So they have 8 legs. They are color-wise, they're kind of tannish, orangish. Usually, there's some really neat SEM, microscope images of feather mites that are painted orange, because that's about the closest color that we have, that I have seen on birds.
Are their legs usually long, short? Do they look like ticks? Like
they're not as symmetrical as ticks are. They have kind of more shape to them, and their legs can be really diverse. Like some species of feather mite have really almost like claw-like front legs to hang on to certain parts of the feather or bird body.
And is that because the bird's going to be flying and they need to hang on? Is that?
Yeah.
Okay, that makes sense.
And there's even been some research about mites that live on the left wing versus the right wing.
Oh my!
And they're whichever side they're on, one of their legs is
that's so cool.
Their left side of their body is longer than the ones on the right side. The right side of the body is longer.
When they meet each other, they say, "I see you live on the left side of the body."
So there's a lot of diversity. They have a lot of hairs, little setae all over them. Those are kind of their sensory organs. large chelicera for their mouth parts.
Like a spider?
Like a spider, yep.
Okay.
Two segmented body.
Oh, a two-segmented body.
Yeah,
okay.
it's a basically the head, what we consider the head, and the body, and that's it.
The cephalothorax?
Yeah.
So kind of like a spider.
Yeah.
Okay. All right. That makes sense.
Very
Like a tick. Yeah. All right. So that's good. So people have a general idea.
Yeah. They and feather mites. I went to a mite conference a couple years ago, and when I told people that I studied feather mites, they were like, "Those are the most beautiful mites. I'm jealous.
Really?
Those are the prettiest mites of all the mites in the world." So
The people at the conference.
yes, the other mite people.
The mite people.
God, I thought you were just talking about regular people, and I'm like,
No.
I think they were just being nice. Like, you probably had no idea what you were talking about.
I'll tell you where my bias goes when I'm trying to think of what a feather mite looks like. I get it wrong in two different directions because I'm like, I kind of picture a tick, which is wrong, but then I kind of picture like a water bear, which also is wrong. But I'm
It's
like,
kind of like
sure.
it's kind of like a hybrid of those.
Yeah.
Yeah.
Wow. Now, one thing that I did come across that I thought was so cool is that a lot of the species have co-evolved, and they live specifically on certain species of birds.
Yeah. That kind of broadly they do. Like certain families of mites live on certain families of birds, and even to some species level, there's only some species of mites that are associated with only one species of bird. But that's not always the case. There is a lot of host switching
Okay.
that goes on.
Oh, Steve, stop.
Uh-oh.
Phenomenon going on right there.
What is it?
I can't remember what they are.
Those to me look like the insect that carries the beachbark disease.
Oh.
I think. I could be wrong. Steve's going to take a picture.
I'll do you one better.
Oh, he's going to iNaturalist it.
No, I'm going to take a video.
Oh, okay. You should iNaturalist it, though.
I can. I'll get a picture.
Eat one, Steve.
Yeah.
For science.
Oops, did I just kill one? It's fine.
There's
Oh, yeah.
They're scattered
all over
over.
the place.
So, folks, there's these little white, fuzzy insects.
And they're just sort of swaying with it. It's not the wind because how much they're swaying, but
They're purposely swaying, just kind of gently back and forth, as if they're almost like they're trying to get our attention.
we have a little group of them, but they're also everywhere, as Alex just pointed out. There just happens to be a kind of a clustering of them.
on this one leaf.
Yeah.
Too.
Yeah, it's just that it's like they love this leaf for some reason.
Yeah.
I don't know.
But there are, I mean, these are beech hulls from beech nuts.
Oh.
So, and I think that's a beech leaf that they're on.
Hmm.
So,
Do you know those little poppers that you throw on the ground? Those little
That is
they
what it looks
explode.
yes,
Yeah, it's like that, but they look fuzzier and smaller,
Yeah,
and they're
those-
just waving in the wind.
white little snaps you throw and
Yeah.
pop on the ground. They look like a miniature version of that. But
Yeah.
not feather mites.
I was hoping we'd know what they are, but I probably should, but I don't know.
We'll put it in the episode notes.
Yeah.
I'm pretty sure that they're the beech scale insect.
Okay.
So,
Oh, yeah, they're all over. Look at that.
let me get in the middle.
Crushing them.
All right, so we were talking about coevolution, and
Yes.
yeah, that some of them, many of them don't have to be on certain species.
Yeah. I think still like at a broad scale, like families of mites are associated with certain families of birds. There's not a ton of huge host shifts from
Yeah.
like different orders. There's some instances of that, but for the most part, they're co-evolved to different groups of birds.
Because as I was reading that, my lack of knowledge of feather mites, tell me if this is totally incorrect, but it just was fascinating to me that you could theoretically take a mite off of one bird, put it onto a different species of bird, and it couldn't survive.
Yeah.
That's so weird to
That's an experiment we would love to do.
But the feather mite activists won't let you do that?
It's more, how do we get a bird?
Right.
Oh.
Right. So,
I'm just imagining a bird hanging out, like flying around your side of the floor, you know? Okay, I have a question.
Uh-huh.
And I'm answer the best you can.
Okay.
It goes without saying. Are a lot of these like they need to be on a bird, or Or how are they transferred from bird to bird? Can they also survive in nests, or like, do they have to rub up against another thing? Or?
These are also great questions, because these are ones that we're trying. Well, for some of those we're trying to figure out. So they are obligate symbionts, meaning that they have to. They don't survive well off of a bird, not for very long.
Okay.
So there's that, but there's been some people that have looked for them in nesting material and stuff, and not really found much. So the transmission question is an open one. Is a huge open one.
Really?
Wow.
Yeah. Well, they're transmitted like from parent to offspring at the nest. Typically, that's like their primary mode of transmission. So like either when the parent is sitting on nestlings, like brooding on nestlings, young nestlings, maybe the feather mites jump from the parent off to the bodies of the offspring and just kind of sit and wait until the feathers develop. Or maybe when the parent goes back to feed the nestlings at the nest, they jump at that point from the parents to the offspring. So primarily their transmission is vertical that way, and that's also why there's this the hypothesis is that there's coevolution, because there's vertical transmission within a species, but we know that there's some species of bird that harbor the same species of mite that are not necessarily this way. So we know that there has to be some level of horizontal transmission across species.
Oh.
Or they've just maintained gene flow for so long somehow, but how are they maintaining that gene flow?
So is this still an area of active research too?
Yeah, this is one of my big questions for my my graduate research and further.
Nice job, Steve. You're asking all these questions that the scientists themselves are asking.
Yeah.
Way to go.
Tell me everything there is to know about cowbirds and how theirs.
That's another good question.
I was just thinking, so I bring up the cowbird because it's not raised by cowbirds, it's raised by other species.
Right.
So in terms of maybe cowbird specific mites aren't a thing. I don't know, maybe they are.
This is a really interesting story because and one that we did a little study on just recently. Well, it was a long time ago, but we're just now kind of writing it up. But so.
Oh, you're writing it now?
Yeah.
Is this a manuscript? Preview?
This is a preview of a manuscript.
Wow.
Yeah.
Cool.
But cowbirds are, people are right now. It seems like brood parasites and mites and lice and all of these kind of host-associated symbionts people are starting to wonder about cowbirds and other types of brood parasites like cuckoos and in Europe and stuff. So, but anyway, cowbirds do have their own specific mite species.
Whoa, okay.
Adult cowbirds do. Sometimes adult cowbirds and juvenile cowbirds harbor non-cowbird specific mites, suggesting that they've at least acquired them from their foster parent. Whether or not those can survive and establish a viable population for long periods of time and potentially transmit to their next host, unknown. But
wow.
It's a really interesting question.
Yeah, because this is I'm trying to wrap my head around this. Steve, just give people a quick explanation. Like cowbirds, nest parasitism, what is it?
Right, so adult cowbirds, when they love each other very much. Well, okay, let's skip that part.
Skip that.
Yeah, they lay eggs in other species' nests.
They don't raise their own young.
Yeah, they don't. And then the cowbird will actually hatch, I think, a little earlier, or they'll kick the other young out, or the eggs before they hatch. And then it kind of grows up, and it gets taken care of by another species until it's ready to fledge, and it's out of there to do the same thing to some other nest.
So, Alex, how can you have cowbird-specific feather mites if the young aren't raised by cowbirds?
Great question.
It just blows my mind.
Yeah, it's the hypothesis. Some of the hypotheses are that the cowbirds aggregate during the non-breeding
Oh,
season.
okay.
But again, where did that first cowbird mite come from?
Right,
in the first place. But the suggestion is that there's horizontal transmission between adults and juveniles. So it's not. I mean, it's not really vertical because it's not at the nest, but it's still within a species.
Maybe the feather mites are bird parasites, and they lay eggs on other birds.
Wait a second.
I think the mite's in the egg, developing with the bird.
Wow. That's so cool.
It's really fascinating.
So this is one reason I love that we keep doing the podcast, because I've had people tell me, we were talking...
You did everything. You've covered every subject
right.
there is to cover.
Well, we were talking before we turned on the mic, folks. Steve and I have been doing this 10 years this month. And I've had people say, are you going to run out of topics? And here's this huge topic that's so cool and so interesting, and Steve and I both know nothing about it.
Yeah.
When he said feather mites, I'm like, oh, okay, feather mites.
You know what? Last episode, we joked about, I think it might have been off mic, we joked about redoing every episode we've ever done just covering the stuff that we skipped the first time around.
Right.
And then we still wouldn't have exhausted those topics. So
it's a beautiful thing. It really is.
It is.
Yeah.
Yeah. Wow. So I have to ask, what do people say when you tell them you study feather mites?
Non-feather mite people.
Yeah.
They usually get, besides that they're beautiful and the most beautiful mites in the world, most people are like, ooh, that makes me itch. That's like the number one thing that people like to say.
All
or
right.
they're like, what? What did you say?
Now, we hinted before that, people are actively researching, are feather mites harmful? Are they just kind of along for the ride, getting what they need, not harming the birds? What's some evidence that they're harming the birds?
Yeah, so the primary evidence that people, with respect to feather mites specifically, are looking at are the presence of feather mites and versus the absence of feather mites and metrics of health.
Yeah.
So could be, immune response or body mass or reproductive output or something like that. And if the presence of mites correlates with the decrease in body mass or decrease in reproductive output, that suggests that that mites could be potentially causing harm. Or if the abundance of mites is correlated in a negative way with those metrics, then that's another suggestion that maybe this is a proxy for a parasitic relationship.
And is there some evidence for that?
Some, not as much as everything else, but there is some evidence of that.
It's not crystal clear.
Yeah.
Is there any evidence of mites affecting feather efficiency, or they're affecting their ability to fly, or
not that I can think of off the top of my head. With lice, there definitely is, because there are lice that eat feathers, like keratin. And those obviously cause harm to the bird because they are
they're taking away some of the feather structure.
Yeah, exactly. And so those have a more direct, clear parasitic effect. But mites are not necessarily doing anything to the feather itself. Some people have wondered, are they adding weight to the bird?
Well, that's what I was wondering. Yeah, because if there's a lot of them, is it affecting how well the birds can fly?
Yeah, and I've seen birds that are just completely covered in feather mites. Most of every feather is full of mites, and it doesn't seem to. Depends how fine tuning, how fine scale of a measurement you're taking.
Right. But I would think it would be evolutionary beneficial for these feather mites not to be harming the bird too much, because it's not like they're a parasite that has another stage where it's trying to get to something else.
Right.
The bird's it, right?
Yeah.
Yeah.
They spend their entire life on the bird. They lay their eggs on the bird, so they hatch. They go through all the developmental stages on the bird. Eat, sleep, reproduce on the bird as well. So that you're right in that it would be kind of against their own benefit
Not that has ever stopped that from happening,
Yeah.
in the natural world.
Right, it's
but
like if they destroy the wing, just sell the wing to someone else and move to another wing.
that's right.
Right.
Yeah.
Like that's what we're gonna do. Right?
Yeah.
So what
I hope that came through.
is there any evidence of like benefit?
So one of the studies, the one that Steve went to, the talk that I gave last semester, I have been studying what feather mites are eating off of feathers. So, like I said, they eat different microbes, maybe oils, plant matter that's living that's stuck to the feathers as well. But we looked at the specific taxa, or the specific species of bacteria and fungi that were inside the bodies of the mites. So what they were actually eating versus
Hello.
versus what was found on the feathers. So like what was available to them.
Huh. So how are you doing
yeah.
that? Are you doing like would it be like an eDNA technique or because you're not actually opening up their guts like I would do with like a fish larvae and like actually see what's in there, right? Like
right, we're using metabarcoding to look at what's on the inside of the mite, so we kind of clean the outside of the mite. We're not digging into their guts because I don't. That would be amazing if we could, and maybe someday we can. But they're so small that it's hard to do that. So just what's
nano machines doing it or something.
Yeah, it's just what's on the inside of the body of the mite versus what was on the feather. So kind of what was available to them on the table, for dinner, and what they actually chose to eat.
So I'm sorry.
Yeah.
Forgive my ignorance. So I'm trying to understand what you're saying.
A bit like metabarcoding.
Yeah, metabarcoding. Yeah. So how did you find out what they're eating?
So we use a technique, a sequencing technique that is targeting a gene that bacteria have, and we also targeted a gene that fungi have. So we extract DNA from the whole body of the mite or the entire feather, and then target sequence that specific gene, and we get back data that's just basically a list of every type of. We have to do a lot of processing to it, but basically a list of the different species of bacteria that are found in that sample.
Okay.
So before all of this, someone probably created a panel for a panel of genes that have specific markers that are specific to certain species. So if you find that gene with the barcoding technique, that's like evidence that species is present.
Yes.
Yeah.
Yes. And 16S.
You can tell him if he's wrong. That's okay.
No, that's right. 16S is the gene that we use for bacteria, and it's universal to bacteria. So any bacteria that's in whatever sample.
That's the ribosome, right?
Yeah.
Yeah.
Yeah. And ITS is another gene, mm-hmm. just like.
That's a huge one for,
yes.
for genetics people. So when you see like species trees or anything like that, back in the day, they were all made with like the ITS sequence. That's just like an easy thing to pull out. And
CO1 is another common gene for animals. So like if I'm sequencing my mites to study their evolution, I'll look at CO1, which is a mitochondrial gene. So it's in any species that has a mitochondria. So we have we can sequence our mitochondrial genes. But for bacteria, 16S is a really common gene to target to say what's in the sample, what type of bacteria in the sample, because any bacteria is going to have that. That's pretty cool. It's going to have that.
Wait, hold on. I see smoke coming out of Bill's ears. Bill, mushroom.
I believe that is American jack-o'-lantern mushroom. So, and the only reason I know that is because I went on a hike with people who know mushrooms. So,
nice.
Folks, we're looking at a conglomeration of medium-sized orange mushrooms at the base of a tree. So
it looks like someone had fun picking some. Maybe.
Yes. So they may be edible. But yeah.
So I was trying to give you something to grasp on too, with all the ITS and 16S.
So basically, just little markers inside of a genome that we can target and figure out what species are within that sample.
Yeah, and the metaphor of the barcode is it's a good one because it's just a little thing that we can scan that will give us the full identity of the thing we're scanning. So it's something that should be unique to that individual species or whatever, that just easy to pull out to identify. So we're what's the book we use for bird banding?
Pile?
Yeah,
Yes.
it's like Pile, but with little genome sequences
Yeah,
instead of looking at retrosea length or
Even
the
more
ratio.
boring.
Yeah. What? How dare you say bioinformatics is boring?
So,
But with all of those data, we looked at, you know, the species of bacteria that were inside the body of the mite versus on the feather, and did some math to figure out what are they actually choosing to eat out of what's available for them. And we found that there's a number of cases in which they are picking keratin degrading bacteria and fungi, you know, picking that to eat off of the feather, which suggests that they're providing like a cleaning service to the birds.
very cool.
That's
Yeah.
awesome.
Yeah.
Because obviously the feather's made out of keratin, right? As far as I'm aware.
Yeah.
So
wow,
what-
that's cool.
so
So,
what
it's
you're
non-random selecting for potentially harmful bacteria.
Yeah.
So what Alex is saying is that there are good feather mites and bad feather mites, like the heroes and the villains.
Every episode we do a breakdown of good and evil. And we,
Yes. We're just kidding. Folks. So there's evidence of some harm, there's evidence of some benefit, and then more evidence of just the,
neutral.
yeah,
Yeah.
good for the mites, but not necessarily harming the bird.
Yeah.
Wow.
Exactly.
All right,
cool.
very cool.
And another thing I thought of when we were talking about the weight, the potential additional weight that mites are putting on the bird by living on the feathers, something really interesting that we've just seen by sampling a lot of birds in the field is that the number of mites on one side of the body is almost always not identical, but very close to the number of mites on the other side of the body. So there's some sort of balance between the two sides. Like if we find one mite on the P8 on the left side, you can look on the other side, and I bet you there's one mite on the other side.
So another one tries again. Sorry, dude. We're gonna make this bird lopsided if you climb on here.
So if you see a bird like
Spiraling
through the sky,
Too many mites on one side.
like it's in like an air show or something.
So I have to ask, you're sampling birds in the field. How do you do that?
Oh yeah, so that's really fun. That is, we primarily use mist nets.
A what?
Oh, Steve.
These are very fine mesh nets that kind of look, to me, I describe them as like a volleyball net, a really wide and tall volleyball net.
A little saggy. I
Yeah,
Yeah.
with some sag to it, yeah. And strung in between two poles. And for a lot of my work, I'm targeting specific species of bird. So I use a method called call playback, where you put a speaker.
Oh.
How
by
dare
the
you?
net. I know. You put a speaker by the net, you play the call of the bird that you're trying to target, and it gets agitated thinking someone's in its territory,
Yeah.
and it flies towards the sound and inevitably gets caught in the net.
How well does that help with like your bycatch?
In terms of like
Like getting something you didn't want to get.
we
Birders.
don't yeah. We don't get too much bycatch
A
that way.
prothonotary warbler. I heard it. Count it.
That is one of my study species, so
Yeah.
I
Oh, wow.
target those.
We can tell you where some are.
Me too. Me
Okay.
too.
Wow, you think you have all the secrets.
I know.
Well,
I don't.
I think I caught every prothonotary warbler in Western New York
Oh
this
my
summer, so I'd love to know
Nice.
if there's more.
Okay.
'Cause
All right,
need
we got to go
more.
out with you more often.
But yeah, so sometimes we put we do the targeted netting. A lot of times we'll use the passive netting as well, where you just leave a net up for about 15 to 20 minutes or so, and inevitably, certain times of the day, birds are more active, and so you will most likely have a bird that just flew into the net passing by. So then we carefully extract it out, and then we measure and do all of our processing, and then we let the bird go.
Do you ban the bird?
Yeah. So we banned.
I was going to say, if you're catching birds and you're not banning them, the bird bander in me would be very offended.
No, we banned all of our birds.
Okay.
We some of them we color banned as well if we want to.
You monster.
If we want to ensure that we don't have to catch them again the next year, but we want to monitor that individual from year to year.
Mm-hmm. So just for the audience, because I'm sure some people don't know, when you color ban them, why don't you have to catch them again?
Yes, so we use different combinations of color bands in addition to the silver, aluminum, USGS band. And so we can look up and say, okay, that bird has a blue band over a red band on its left leg and a USGS band over a yellow band on its right leg. I know that bird is Bill that I caught last year.
Right. So it's a way almost to give the bird a name. You're giving it a code so you can look through your binoculars and know what bird it is. Yeah.
Exactly.
Okay.
Yeah. And we do give our birds names. Very,
you got to give us a name
names.
now. We need to know what's the one you wouldn't want someone to know.
Oh no.
And don't worry, it's only on a podcast that we're recording. Fine, you can give us a couple down the list that maybe not the worst, but the.
We did. I was just looking through our data yesterday, and one made me laugh. There was a porcupine on the side of the road that was dead, right by where we stopped to catch this bird. And I almost said it wrong here, but in my head I said, my friend was sitting next to me, I said, "Oh, look, a porcupine." And she was like, "What?" And so we named the next bird Porcupine.
Love it.
And I'll never forget what that bird was, where we were. You know, it's a really good way to remember what was happening at that time.
Beautiful.
Good old porcupine.
Good old porcupine.
And then with feather mites, you know, we just had, on our last episode, we had Jason Dombrowski, who's the head of the Cornell Insect Lab.
Okay,
I bet you guys would hit it off.
I bet you would.
That's awesome.
But his specialty is micromoths.
Oh,
and he said, you know, to identify micromoths, you usually need to look at their genitalia.
Yeah.
So how are you identifying feather mites?
Yeah, genitalia is one. It is one way. So they have. I mentioned that they have a lot of CD in hairs all over their bodies. So sometimes some of the diagnostic features are the numbers of hairs on certain segments of their legs. Or the distance between certain CD on their backs, or the length of their reproductive parts. So there's
using a number of different things.
Yeah, a number of different things, a number of different morphological features. Overall size is one of them too, but they're very, as you can imagine, very fine scale measurements.
You're not eyeballing most of these things?
Not eyeballing most of them. No.
Maybe are there some you can't tell apart? Like, so I'll just say, coming from the plant world, I was at this aquatic macrophyte workshop where some experts who had actually written the key on aquatic macrophytes were kind of letting us, teaching everyone how to identify. And they're like, if you come across this one, you actually can't tell it from this other species. But maybe if you're in this area, you can just assume it's, Elodea canadensis, but if you're not, then just put the genus down, you're fine. Other Potamogeton actually also look just like it too sometimes, so sometimes you can't even get the genus. And that's with plants. That's with stuff where we can grab right out of the water,
Right, yeah.
So.
I think it depends, but if you're looking at species of bird that have never been studied before, their feather mites have never been studied before, you see a species that you can't key out, which has happened to us. It's like, okay, it doesn't follow this, it doesn't match this, it doesn't match this, it doesn't match this next piece in the key. Well, it's probably a new species, which is pretty exciting.
Have you heard of iNaturalist? You can take a picture. It'll identify.
And I'm sure a bunch of amateurs are going to jump on there and be like, I know this one. Well, okay, so as a guy who does a lot of genomics, we typically need a certain amount of material of the species to take the sample of and then extract the DNA from. But when it comes to mites, you have a few thousand that you happen to know are the same species. Grind them up. No, what do you do with? Because this is a world that I have no experience in whatsoever.
Yeah, that's a very good question and something we have struggled through.
Sure.
But so there's a couple different strategies. You can sequence DNA. I mean, you can sequence a couple of genes from an individual mite. It is possible. There's a couple of tricks you can do when you're starting the sample. You can poke a hole. So I've poked holes in the exoskeleton of mites to help release the innards in the DNA.
How often in your experience, and this would, as a fellow scientist myself that does lab work, this wouldn't reflect on you as a scientist if you if you admit to this happening. But how often do you do an extraction and be like, well, there was nothing there, we have nothing, It
happens. Well, it happens a lot, but I still try to sequence it, and sometimes it works.
Okay.
So, for my master's project, you have some evidence, with gels. We can do an agarose gel, and if the band is there from our amplification, we can say, okay, it's probably going to sequence. So we send it off anyways. But for my master's project, we got I thought everything worked, and then I got the data back. Half of them were human DNA, which I'm assuming is my DNA. So it just, you kind of have to plan for about half of the samples not amplifying.
Okay.
But with genomic work, so that's just single gene amplification. Genomic work, we came up with a strategy to do a pooled sequencing approach, where we take all the individuals from, or a certain number of individuals from a single feather that we can identify under the dissecting microscope as the same genus, because that's as far as you can get down to under a dissecting scope, and just cross your fingers that they're all the same species, and like you said, grind them up.
You weren't too far off, Steve, right?
Yeah. But we did we did do whole genome sequencing from a couple of individual mites, because we were like, why not? Let's see if this works. And we got one to work really well, and we came up with a draft reference genome for it.
I think I saw that in your publication, that you had a draft genome out there. Yeah.
That's the one.
Very cool.
The one. And that is actually a really fun mite species that has the draft genome. And you were asking about this, more or less the specificity and the coevolution earlier. This mite and Prothonotary. So this mite is the only mite species that is its only known host is a Prothonotary warbler. So it's specialized to Prothonotaries far that we know.
I know sometimes with maybe this is more about parasitic relationships, that sometimes you will find some horizontal gene transfer. Is there anything known about that with mites and birds, or?
Nothing's known about that, but that would be cool to look at. Yeah. So Prothonotaries have a special place in the feather mite world because I did my graduate work in Arkansas, and they are abundant there.
Wow.
So a lot of my projects involve Prothonotaries and their mites. So we have a lot of data on Prothonotary mites.
Any branching out into any other warblers? They're a favorite of a lot of people, I guess. So.
Yeah. Besides Prothonotaries, the rest of my dissertation and a lot of my master's work was on just all warblers.
Got it.
So we sampled almost every species of warbler that breeds in North America.
What? Holy cow!
For feather mites.
Wow.
To try to figure out, okay, what is their actual specificity? Let's go out and get all of these birds that are within this family to try to really measure that level of specificity that they have to their hosts. And Prothonotaries, they only had that one mite was only associated with Prothonotaries. There's one mite species that's on 17 different species of warbler. So it's a really very different, ecologically than the specialist that's on Prothonotaries. And the only species of warbler that didn't have any mites, we found no mites on any of the warblers in the Southwest. So Lucy's warbler, red-faced warbler, painted redstart.
Any idea why?
I'm assuming it has to do with the
environment.
Yeah.
The dry heat.
The dryness. Yeah.
So I don't know if you guys looked at this, but is it like if you were to look at the relationships between the warblers, is it more about geography or is it more about closeness and relationship?
I think it's geography, because some of those species out in the west are the same within the same genus.
Got it. So there was losses, based on geography. That's really cool.
We went in a drought year too, which maybe that had something to do with it. So I'd like to go back and do some more sampling and see if there's. I doubt anything would come up because we sampled quite a few individuals of every species, but.
Are people okay? Have people done? I say people. This could be you. Have they done like time series? If they're capturing the same bird, are they then re-evaluating the mites over time?
No.
No.
That would be cool. That would be a good thing to do. To look at. We've talked about that a little bit with some experiments that we've done. Like, it would be really neat to see, is this population the same over time?
This is, in a sense, this is a very exciting topic to ask questions about, because it feels so young and burgeoning that, like, it's sort of exciting right now, and there's, like, it sounds like there's people are actively doing this stuff.
Yeah.
Or writing grants to be able to do this. So.
Alix is here at the dawn of feather mite science. Right?
Well,
she's a pioneer.
There's been I mean, feather mite biology has been going on since. At least, like, I'm trying to think of some of the older papers that I cite from the 1950s. Those are some of the older ones, and some people were doing taxonomy and stuff obviously before that.
There were literally like 5 people across the U.S. in previous years.
I think I was just talking to one of my colleagues in Spain. He's another feather mite researcher, one of the very few. He was like, I think you're the only person in the U.S. now that's studying feather mites.
Seriously?
Yeah.
Wow. She came on our podcast?
We have the feather mite person of the Americas.
Well, thank you.
When you're someone who publishes and sees someone else who publishes, sometimes you're like, I wonder, how they compare. Alix has way more publications than me, and their H-index is higher and all this stuff. Is part of that you're part of that? Well, that one, you're an impressive scientist, of course, but another thing is like that you're one of the few people doing some of the things you're publishing. I think
that has a lot to do with it, because we can ask.
Make me feel better about my H-index, please. No, okay.
No, I mean, we have the ability to ask like really simple questions and really complicated questions within the same system, and so we can we've been able to publish kind of like fun little notes that are just like, this is a really cool thing that we saw, or we have a little bit of data for this, but it's nothing is known about this, so let's go ahead and publish it. And working with warblers is really fun, so getting to go out in the field and do lots of experiments with those allows for that kind of, you can collect a bunch of data for multiple projects at the same time too. So that's kind of, I think that has a lot to do with it, and the fact that no one else is studying this, so it's pretty.
It's open territory.
It's open territory. Yeah, I think that has a lot to do with it.
So that's actually a good point, because we're kind of at our point here. So
okay.
Is there a place where people can find your research and look into your research and find out more about what you're doing?
Yeah, all of my papers are on Google Scholar, but I have a website that I think is pretty. If you Google my name, you can find it pretty easily, and I've got some anecdotes about the projects that we work on, and a list of all my papers, and a little bit more about me and that kind of stuff. So.
All right, so
you can also give the address now if you happen to know it, and people while they're driving can type it into their phone.
I think
we always encourage that on the podcast.
I think it's matthewsalix.weebly.com.
Okay.
I think I have it backwards, but maybe it's alixmatthews.weebly.com.
Try both.
They got all the time in the world. No, we'll put it in the episode notes as well.
Sounds good.
Steve's ducking under some Norway spruce here. Let's try to take him out.
Uh-huh.
Well, Alix, I have to say thank you so much for agreeing to be on the podcast, but also for
Getting berated by questions and taking it like a champ.
and being so tolerant of our constant interrupting of
I love it. I love
You were accosted during this episode, and I'll admit it.
But no, it was great. And maybe we can have you on some other time, and you can tell us about other research and talk more about birds.
There's plenty. There's plenty.
All right. Well, thank you again.
Yeah, thank you.
All right, folks. Steve and I will record the wrap-up at a later date, probably, right?
Mm-hmm.
All right.
And this will be, this is going to be the October episode.
Right.
So
spooky.
It wasn't the spookiest of episodes, but maybe people will be a little bit fearful of thinking they're covered in mites now.
There's spiders, spider relatives involved.
Yeah. So.
All right, folks. So stick around for the wrap up.
All right. Steve here. Happy Halloween, folks. We hope you enjoyed the episode. And sorry about the beeps that you're about to hear. I just couldn't figure out how to turn them off. First and foremost, we'd like to thank our growing list of Patreon supporters. So thank you Samantha B. and Deborah B. We'd also like to give a special shout out to our top patrons at the end of the episode, so stick around to hear Bill's daughter Violet share that list. And if you would like to support us on Patreon, you can get access to special patron-only versions of the episodes that include Bill sharing the episode notes. And maybe I should start sharing the notes too, pulling my own weight. All right, well, because of our support from patrons, we're able to keep the show free and do cool things like maybe take a trip out to visit Daniel so he can do an episode for us like the good old days. I don't know. But you can also make a one-time donation through PayPal on our website, or help out by sharing the podcast with friends or family, and by subscribing to the podcast and leaving us a review wherever you get your podcasts. It helps spread the word and helps us reach a wider audience. Speaking of which, I'd like to thank our newest 5-star reviewer, Sloth Travel Club. So come enjoy our sporadic posts on Facebook, Instagram, and Blue Sky. And you can always email us ideas for episode topics, criticisms, or even your own encounters with feather mites. Only feather mites, no other mites, to thefieldguides@gmail.com. And parents, remember to get those kids outside, let them get muddy and dirty and flip over rocks and logs. And if you don't have kids, find time to get outside yourself. And here's Violet to share our top patrons.
All right, and I'm here with Violet, who is putting off getting ready for trick-or-treating for maybe the last time this year, right?
No, not going to happen.
All right, here is Violet with this episode's top patrons.
Eric, Alyssa, Adam, Mary, Colin G., Dr. Judy, Kimberly B., Peter R., Callie, Rich K., the Hebranks, Orange Julianne, Rachel L., the Drunk Vitologist, Daniel M., Diane, We Named the Dog Indy, Hot Dog Cold Weather, Dwayne H., Jennifer D., Jonathan A., Deborah, Samantha, Jeremy M., Licky Weeks, Lori B., Michaela, Val, Andy W., Elizabeth P., Picasso 3, John R., Evan, Mark T., Cecilia, Julie B.,
Take a breath.
Dennis, Chris N., Sung, Jeff S., Jonathan K., Matt E., Plants in My Pants, Ryan R., Sophie S., Connor H., Measure and Principle, Fragaria Pepaloinoidea, Outside Chronicles, Andrew C., Quixote, Max D., Melissa Marie in Dusty, Arizona, Kelly S., Sarah S., Helen A., Judy F., Ben C., Jane H., Doodle Dude 82, Gallon Mac, Kazis, Jeff S., Esther C., John W., Mark V., Bethany H., Rob M., Holly M., Catherine W., Virginia A.
Well done. We can go trick-or-treating.
Okay.
All right, patrons. As always, thank you so much for your support. It really means so much to Steve and myself. Every episode, when we read this list, I'm just blown away with gratitude for your support. So thank you. We will continue to try to live up to it. And everyone, happy Halloween.
Goodbye.
I meant to say this while we were recording, but anything bigger than a mite has mites.
All right, so Steve has mites.
Wow. Well, is it true?
Yeah.
Okay, it's true.
Yeah,
okay,
yeah.
Got it.
And you do have mites.